I think we're ready to get started with the next session. So our next session is called Current Immune Mitigation Strategies for Gene Therapy in Seropositive Individuals. We have Carsten Bonnemann from NIH who's going to kick us off with a talk on intrathecal gene therapy for giant axonal neuropathy, immunological considerations and observations. So.
Thank you. Thank you to the organizers for putting together such a great symposium and workshop. And thank you for including us in the presentation. So my disclosures are, oops. This doesn't advance. Thank you. So I'm a PI on this a trial which you'll be hearing about, and it is sponsored by NINDS at the current time without any commercial participation at this point. Just briefly about giant axonal neuropathy. It's a neurodegenerative disorder of early childhood. It's an autosomal recessive disorder with early onset of initially sensory neuron, then motor neuron, and then central nervous system degeneration that is relentlessly progressive over the first decade and two decades of life. It's a loss of function of gigaxonin, which is a ubiquitin ligase adapter protein that's necessary for the breakdown of intermediate filaments. And as a result of that, you have backup in intermediate filaments in any cell of the body, but really mostly in neurons with long axons, which swell up because of the backup of the neurofilaments and giving the disease its name, giant axonal neuropathy. But other intermediate filaments are affected as well, including keratin in the hair. That's why the children have this curly hair phenotype that is so distinctive. Because it's a small gene and is loss of function, it really is a great candidate for AAV-mediated gene replacement therapy. So how do you get to the CNS, to the relevant nerves? There are different ways to get there. And we chose the intrathecal route, the lumbar intrathecal route for this particular trial. And this, by the way, was the first ever delivery of AAV into the CSF directly in human. So it came together by really a team that was instigated by Hannes Hope Fund, Norris, which is a patient advocacy organization, and there was an affected child, obviously, in the family. And she reached out to Steve Gray at UNC at the time, now UT Southwestern, to create a gene therapy vector that would be applicable to this disease, including generating the preclinical data and the IND-enabling data for that. And then there was, of course, no support or funding to do a clinical trial, and that's when they approached us at the NIH to design a protocol, design a clinical trial for this application as the first intrathecal trial of an AAV product. And that led to this trial here, which was a single-site, non-randomized phase 1 dose escalation trial in children with GAN 5 years and older. You can see here this amazing team that came together, and my institution, Diana Brocha-Goebel, Josh Todd, and Dima Sade were leading, and Steve Gray, of course, is behind all of this. I should say there was industry participation on and off during the trial. At the moment, not, but all of these were helpful partners in this trial as well. This is the agent. It's a self-complementary. It's small enough to make a self-complementary product in AAV9 with a ubiquitous but weak promoter that is driving a codon-optimized version of GAN. There was a dose escalation from 3.5 E13 vector genomes per patient, so it's not per kilogram, per patient, to the 10-fold higher dose in 4 dose steps given by intrathecal administration in a Trendelenburg position with a 1 hour post-dosing period where the patient was kept in the Trendelenburg but rotated to allow percolation of the vector along the neuraxis with the goal of hitting the targets predominantly in the spinal cord, but of course, if possible, also higher up in the eye and the brain. And this is how it looks like in the actual situation. There was a nerve biopsy, a sensory nerve biopsy done before here. You see the vector going into the patient, and the patient in the Trendelenburg position. This is in the paper just to say that the patients were already symptomatic in this trial, 6 years and older, were dosed, and they already had a clear electrophysiological involvement with absent or reduced CMAPs and sensory nerve action potentials. This is Federico's slide of the phases of immune considerations with the pre-existing immunity, innate and adaptive immunity, and we'll be touching on some of those as I describe what we saw and found in this trial. First of all, the safety was quite reassuring. It was actually very good. The only effects that we saw that were related to AAV really were this CSF inflammation that was clinically silent that I'll be touching upon, and then one fever post-dosing that required prolonged hospitalization. Other than that, all adverse events were really related to intercurrent illnesses or the disease itself. So quite favorable safety profile. And no evidence of DRG toxicity. I'll come back to that as we go forward. Also, no systemic findings, no hepatopathy, no liver function elevations, no TMA, probably because of the low, of course, systemic exposure that we saw. So coming to the immunology, this trial did not exclude patients with pre-existing neutralizing antibodies. And you'll see it had some difference in terms of the antibody response, but the effectiveness of the therapy seems not affected by this. The reason for this decision was data from many groups, but also from Steve Gray, that intrathecal dosing in the presence of pre-existing peripheral neutralizing antibodies is positively still able to allow for transduction of targets such as a DRG. So that highlights this as an immune-privileged space, at least in the presence of naturally neutralizing antibodies pre-existingly. Our cohort was broken down into about half and half seronegative and seropositive patients to some degree. And you saw this slide already from Roberto, who did all the immunological analysis in this talk, and then also Jessica Chichester at UPenn. But you can see these are patients that are seropositive in solid, seronegative in solid, seropositive in broken lines. You can see both have a rapid response in neutralizing antibodies in the serum. Both also have a neutralizing antibody response in the CSF, but they're at baseline all negative in the CSF for neutralizing antibodies. That's probably why this is possible. And you can see the seropositive patients have a somewhat faster response in going up and maybe a little higher initially, but then plateau out at about the same levels. This patient here is notable in that there is no neutralizing antibody in the CSF and a dampened response in the periphery. This was a patient who was seronegative at basis, but was also CRIM negative and received immune modulation. And we'll come back to that later, but it's still unclear why this patient remained negative in the CSF. So the response in the CSF is prolonged and sustained as it is in the periphery, and that, of course, puts some doubt on redosing into the CSF after exposure into the intrathecal space. Vector shedding is also interesting to observe, and we're just going to look at the blood here. These are the different dose groups, and you can see the highest dose group of 3.5 E14 has a prolonged and high persistence of the vector. This is, of course, a function not only of the high dose, but all patients, as you'll see in the highest dose group, also received immunomodulation, rapamycin, sirolimus. So that may have played a role in the persistence of the vector here as well. When you break that down in baseline seropositive to seronegative patients, you can see the seronegative patients had a longer persistence of vector post-dosing in the blood compared to the seropositive patients. And you can see that here a little drawn out in individual patients where you can see the positive patients had a more rapid clearance of the AAV, more rapid shedding of the AAV compared to the seronegative patients who had longer persistence of the vector. What about the immune modulation protocol? So in this disease, we have patients who have CRIM-negative status, which means they have no gene expression or protein expression from either allele, so they're basically immunonaive. And other patients have missense variants where some protein is being made. So this trial was designed to include both CRIM-positive and CRIM-negative patients. But the CRIM negativity in this condition carries a risk predictively. If you deliver by the same route of administration, same vector, same promoter, a foreign protein by the same route of administration to non-human primates, they'll develop an encephalitic response that's T-cell predominant. And if you pre-treat with sirolimus or rapamycin, that response is subdued. And therefore, we decided to design a protocol that would take advantage of this preclinical knowledge proactively to not allow any CRIM-negative patients to go into the trial without immunomodulation, because we were afraid that transgene-naive patients would mount a rejection response that's T-cell driven. And the protocol that resulted from that is shown here. So it's basically a mini solid organ transplantation protocol that's kind of pared down and damped down. So all patients will receive prednisone, pulse first, and then prednisone for a period of 4 months. I'll show you later why that is. And then the patients with predicted CRIM-negative situation would also get rapamycin and tacrolimus, two T-cell active drugs. And we decided at the highest dose group, irrespective of the CRIM status, everyone would get rapamycin, and the CRIM-negative patients would also get tacrolimus. So at the highest dose group, everyone has prednisone and rapamycin, and the CRIM-negative patients have tacrolimus in addition. Why did we extend the steroid to 4 months? So initially, the steroid coverage was only shorter. You can see that here. And with dose escalation in the third highest dose of 1.810E14, in the first patient that was dosed, who was a patient who was at baseline seropositive but CRIM-negative, we saw this rapid increase at 3 months of CSF white count and protein, which was immediately responsive to reintroduction of steroids. We therefore extended the steroid coverage out to 4 months, followed by a taper, and that resulted to a blunted CSF response. All of this was clinically silent, so the patients had no clinical symptomatology of this CSF inflammation. But the extended steroid regimen was enough to suppress that, and it returned all back to normal by 1 year. So that was self-limited and steroid responsive. What about biodistribution in this situation? We only have one patient who tells us biodistribution, who was in the first dose group, lowest dose group, was an NAB positive patient at baseline, who passed away 8 months after dosing from complications of the underlying disease. This was a more advanced patient who passed away, and the family graciously allowed for a post-mortem. And you can see the biodistribution here at that low dose is a gradient, but we do get transduction effectively in several levels of the spinal cord, as well as in the periphery, despite the presence of neutralizing antibody peripherally. And the good news in that N of 1 limited is, but it really reflected preclinical prediction of the biodistribution of intrathecal dosing at that dose. So that was good to see, and not impaired by the neutralizing antibody positivity of that patient. There is also a signal of efficacy that is not dependent on NAB positive status prior and post-dosing. And just one slide on the efficacy that we found there. We used both independent natural history, but also the trial patient's own run-in data to determine change of slope. The primary statistical outcome was the Bayesian analysis of the slope pre and post dosing in individual patients, and the pre-specified endpoint was a 95% posterior probability of slowing of the course. And you can see the post probability densities here. This is a natural decline here in the motor function measure 32. And you can see at the lowest dose, there's no shift of the posterior probability. But as we increase the doses, we're shifting the probability of decline towards 0, as you can see here, with a 99% probability at this dose group here, which you see here. And therefore, the trial met its statistical outcome. And that's shown here in the frequentist analysis, where it's also statistically significant. So meaning we are hitting the endpoint irrespective of the NAB or CRIM status of the patient. And just briefly, of course, the next phase of that trial will be going to less or pre-symptomatic or oligosymptomatic patients, because you can imagine repairing an axon that is this long, almost drawn to scale, is a heavy task. What about DRG toxicity? There was, of course, clinical as well as preclinical reports of AAV associated DRG toxicity, such as in this report here on SOD1 gene therapy done by Bob Brown. And this was a microRNA, where there was a pain syndrome setting in 6 weeks after dosing that required treatment, escalation of steroids. In our trial, we were fortunate to not see that. In contrast, we actually saw evidence for DRG restoration. And so here you can see the natural history of sensory nerve action potentials in this disease. This is stimulating sensory nerves and measuring their response along the length. You can see here in classic untreated GAN, they're completely absent and never return by 9 years of age. However, what we saw in the trial, and that really surprised me and continues to surprise me, is that we saw reappearance even from negativity or increase of amplitude after dosing, and that continues to go on 4-6 years out of the trial. You see evidence of the restoration of the sensory nerve function in the upper extremity, where the nerves are less affected at dosing. We also see emergence of regenerative clusters in the post-dosing biopsy as an evidence for regenerative activity in the sensory nerve. So no clinical and no physiologic evidence for DRG toxicity, but restoration. And then just very lastly, the immune modulation effect on the anticapsid responses that we observed. One is the early spot response, and so you have them broken down here in patients without T-cell immune suppression and patients who received the T-cell immune regimen, either rapamycin with TACRO or just rapamycin. And you can see already from the distance overall, we have less early spot response against the capsid. And the same seems to be true for the neutralizing antibodies. If you these are months, broken down by months, so 3 months, 6 months, 9 months. The red ones are no immunosuppression, no immune T-cell active immune regimen, and the blue ones are with. And you can see there's a trend for the ones under immune protocol, significant here in this one, that there is a lesser NAB response post-dosing in the patients who received the immune modulation. So again, I want to thank everyone in the team who participated to that, and I'm very happy that aspects of this clinical trial that's published here now have now found their way into other intrathecal trials, both with the immune modulation regimen, but also with the dose and administration route that we've chosen. And two of the bespoke gene therapy programs are choosing exactly that platform. So again, thanks to everyone involved, including Roberto and Jessica at the Penn Immunology Core, and an enormous team of people, in particular, Diana Barucha-Goebel. Thank you.
Thank you, Carsten. So just like we did for the last session, we will save questions until the panel. So our next talk is Giuseppe Ronzitti from INSERM, and the title of his talk is Toward AAV Gene Therapy in Seropositive Crigler-Najjar Patients.
Hello, everyone. Can you hear me? Yeah. So let's start talking with the disease. So it's Crigler-Najjar syndrome. It's a ultra-rare disease. You see, it's 1 in a million at birth. And it's a deficiency, a strict deficiency of an enzyme of the liver, of hepatocytes, expressed in hepatocytes. And the absence of these enzymes leads to a jaundice, so an accumulation of bilirubin in the circulation that, if left untreated, can cause severe neurotoxicity and also death. During the early ages, there is in fact a severe risk of neurological death, which is controlled as is due in neonatal care with blue light, so the use of phototherapy. This is a treatment that allows patient survival, but is quite heavy for the patients and the family, because they have to undergoing session of phototherapy, daily session of phototherapy, between 8 to 12 days, to 12 hours per day. It's a symptomatic treatment. Basically, the blue light transforms the bilirubin in another molecules that can be eliminated, excreted. And there is also an issue with efficacy. Of course, there is an issue with compliance, but there is an issue also with the efficacy that decreases over time. And the only really treatment for the disease as of now is the transplantation that is really curative, because this is really a strict liver disease. But still, I mean, it's going to be a different disease in the sense that the patients will be immunosuppressed for a long term. We started working in, once again, it's about Federico. We started working on that in 2013, actually, 2012-2013, when I arrived at Genethon to be a postdoc in Federico's lab. And this project, so we basically did the whole development inside Genethon, and we started the trial in 2018. We also get funded by European Union grants, which actually supported part of our activity as much as AFM Teleton that support, as you know, Genethon. So before I go to the slides with results, the clinical trial that we started was a phase 1/2 clinical trial that included adult patients and that were undergoing phototherapy. And there was, of course, the safety, which was important, but also the efficacy measured as the interruption of phototherapy 16 weeks after the injection of the vector. So you see here the results. I'm just going fast on the results because I want to speak actually about what's come next. But what is important, I think, is to notice here in transparency, you can see the results of the first cohort. And the first thing you can see is that in the first cohort, which is the lowest dose cohort, you see a transient efficacy, which is lost with time. So what is interesting is also Audentes have seen similar results. There are the first patients injected in Audentes trial, which was doing the same trial than us. They had exactly the same profile than our first patients treated. So this we consider this as an effect of really related to the disease, because we have seen that also in preclinical models, and we decided to go to the higher dose. So the other thing you can see here is that the levels of bilirubin, these are patients that are undergoing phototherapy, 8 to 12 hours, and they still have very high level of bilirubin, around 200 to 400. This is the range that we have. And so right after the treatment, a very fast expression of the transgene and elimination of bilirubin in circulation. And so the level of bilirubin goes down to around 100. And what happened then is that given these results, so this is interesting because these are the 3 patients of the second cohort that we injected, so with the higher dose, and they are very consistent. And we were able actually to stop phototherapy at 16 weeks, and then the levels stay low up to 24 weeks. Another interesting thing about the disease, it's about also its clinical endpoint, but it is something that is clearly observable in the patients directly, and they that they are actually changing because the skin and the sclera of the eyes, they are yellow due to the accumulation of bilirubin, and they become basically pink and white after the treatment. So this is one of the reasons why we were not able to actually do a controlled clinical trial, placebo-controlled clinical trial, because actually the patients, they knew basically that they were being treated with the vector. So we provided the proof, clinical proof of the efficient gene replacement in the context of this disease for up to 8 weeks. This is what we published as of now. We started a pivotal phase to confirm actually what we have seen in the first cohort. The interesting thing is that in the pivotal phase there is also pediatric patients involved. And we are convinced, I mean, I think when you see these results, we are convinced that the clinical endpoint that you see here are very robust and they are very reproducible. And we think that this is a very good model to evaluate new strategies to overcome the current gene therapy limitation. And speaking of that, we are right to the, I mean, most of the people have shown results like results about this issue related to the presence of antibody. Of course, this is related to the efficiency. In case of whenever you have a high level of circulating antibodies, you neutralize the vector and then you don't have any transduction. Whereas when you don't have any antibody, you do have an efficient transduction. But what we are interested also, I think, and this is, I think is also important, is that that situation where you have a decreased transduction, so antibody that can affect transduction, not completely, and they can have also an impact on the safety. So in these conditions, I heard someone say, we can, of course, raise the dose. But whenever you raise the dose, combined with the presence of antibody, we don't know what's going to happen because there could be also an adjuvant effect between the dose of AAV and the presence of antibody. And this is particularly related to the fact that you are creating immunocomplexes in a context where you have too much vector, but also the fact that you may have complement activation and then organ-specific toxicity. So for this reason, for both reasons actually, to increase efficacy of the treatment, but also to reduce the risk, the safety risk associated to the administration of high doses of vector, we at the time we thought to use this IDS, or this immunoglobulin degrading enzymes, which is a bacterial enzyme which was actually used in transplantation. So you see this is kidney transplantation. These are kidney transplantation patients. It was the first proof, clinical proof of the use of IDS in patients with kidney transplantation. And you see that right after the administration of IDS, the levels of antibody, they go down well, to very low levels. I cannot say to zero. You will see afterwards what happened in preclinical animals. But they go down to very low levels, and then they come up in 7 days. So this was something, it's a drug, it's called idefirix, approved in Europe for use in HLA-sensitized patients. And we provide demonstration, this is an old story, once again with Federico, we provide demonstration that we can use this to basically transfer a gene in seropositive monkey. But now we come to the new data. We had another question, and the other question was, can we reach basically the same level of transduction in the liver when we compare a seronegative monkey and a monkey which has a very high seropositive titer? So we did this experiment. These are 2 monkeys per each, so 2 seronegative monkeys for AV9, and 2 monkeys with the titer which is quite high. This is a titer that is quite neutralizing for this dose of vector. And we administered the monkey with IDS, and 2 days after we came with an AV9 vector at the dose of 2,5E13 dose. This is a dose which is, you can probably are familiar with these are doses that are more for the muscle. It is the dose that we are using, for instance, in the clinical trial for Duchenne muscular dystrophy. But we wanted to understand what was going to happen in the liver after the use of this dose. And so you see, first of all, how it works, IDS in the primates. This is important because, so this is the Western blot of immunoglobulin. So this is the band compatible with full immunoglobulin. And then there is a small band, which is the single-chain immunoglobulin. So it's where IDES had cut just once. And then you have the FC. So after 2 days of treatment in both primates, it is quite well conserved. You see that there is a very nice decrease in the level of immunoglobulin. Still, there is some residual immunoglobulin, and you have also some single-chain IgG. This is, we have demonstrated that this is typical of primates, and in humans, it's a little bit less pronounced, the persistence of single-chain IgG. And what happened in the transduction of the liver? Here, these are the results of 4 regions of the liver, 4 different regions of the liver per each animal. And so you see in terms of VGCN, we have no difference. I mean, it looks like there is an increase, but in fact, it's not significant. In terms of the vector, which is present into the liver. And the expression, the RNA expression, are also conserved. And still, there is still this small increase, in particular in one animal. So it looks like these levels, which are very high compared to what we use in the clinic. I mean, these are levels, how we selected these levels. This is important because basically these are the levels where in a population of Crigler-Najjar, they will allow us to treat the most of the population. So include basically almost all the patients that are seropositive. It's 3 out of 15 that has levels that are higher than that. So this is important because we demonstrate that depending on the dose, of course, of the vector, we can achieve same expression in the liver. But what's going to happen in re-administration, because there are 2 issues, as you know, about antibody and administration of vector. The first being the pre-existing immunity that you have in primates, and the second being the re-administration setting. So to do that, we did an experiment in rabbit. You know very well, these are selected basically along the years to create antibodies. So this, if you inject them with an AAV, for instance, you will have huge level of antibodies, and you will see that afterwards. So we pre-immunize those rabbits, because rabbits has no natural immunity to the vector. So we pre-immunize them, and we then treated with IDS at day -2 and day 0. So there is just a small thing to say is that IDS works very well against rabbit IgG. So you have a higher level of IgG, but you also have a huge, it's not comparable to the one that you have in humans, efficacy of IgG. And these are, so this is what you can see here clearly, that starting from basically day 1, you have not much IgG. You have some residual FAB prime, which are partially neutralizing, and also the FC that is created right after the injection. So after 2 days, we injected the vector, and these are the results that we obtained. So when the rabbit, they were pre-immunized, these are the anti-AAV8 neutralizing antibody titers, then the rabbit were pre-immunized, essentially they had the same levels of IgG pre and post, because these were treated with PBS. Right after treatment with the IDS, you see that there is a huge decrease. This is how good is IDS in rabbit. I mean, it's really impressive decrease in the amount of neutralizing antibodies circulating. I mean, knowing that here you don't see anything, but here there is still a residual neutralization titer. And this neutralization titer, of course, if you inject a vector there, you will not see the expression, because these are still neutralizing antibodies that can prevent the expression of the transgene. So these are part of these results and other results, in particular, that we collected through the collaboration with ANSA Biopharma. We were able actually to start a clinical trial on IDS, so to apply actually the use of IDS on Crigler-Najjar patients, seropositive Crigler-Najjar patients. And this is, of course, you understand that this is, I mean, for an institute like ours, it's an important opportunity, because we will allow actually to participate to all the patients to the clinical trial, so those that were excluded right now because we cannot include them when they are seropositive. And second, for the data that I show you, I think I'm feeling very positive for the data I show you about the rabbit, although the IDS, as I said, is working pretty well in the rabbit. But I'm feeling quite positive also for a potential readministration, at least in those patients that had that after vector injection they had a lower levels of neutralizing antibodies. And then I finish here just with the acknowledgements. What I wanted is, of course, the team. In particular, I wanted to acknowledge the funding agency. These two projects in particular, we are still working on the optimization of ideas, and then we want to achieve like a better efficacy in preclinical models, but also in humans through the engineering of the IDES enzyme. And I finish with that, yes.
All right. Thanks, Giuseppe. So up next we have Barry Byrne from the University of Florida, and he'll be talking about AAV redosing.
Sorry.
Disclosures. I like to use this to represent really what happens after systemic exposure. So I'm going to be talking really almost exclusively about that. Obviously, we've heard a lot about both the innate and adaptive acquired responses, but I think we have to think of that in terms of what's happening in the liquid phase in the blood. So it may not be thought through for everyone that's been doing this in mice, but in humans where you can take regular blood samples after the exposure, which we now routinely do at 1 hour, 12 hours, and 24 hours after the infusion to understand the early kinetics, and then daily for the first 14 days at least to understand all of the events that occur both in the antibody PK, the vector PK, and the immune effector events that follow infusion. But after a typical 1E14 to 10 to the 14th vector genome per kilo dose of AAV delivered systemically, about an hour after the infusion, the vector concentration in the blood is about 10 to the 11th per mil. So it's a massive exposure of the capsid proteins, and their surface area is not to be discounted because it's about 25 meters squared in a typical 40 kilo person, which is about half of the lung surface area. So this itself is something that we can't change entirely about the properties of the vector. So that initial exposure injures endothelia and contributes to some of the innate and ultimately adaptive responses. And of course, pre-existing antibodies play a role with that, but interestingly, 1 hour after an infusion, all pre-existing antibodies, no matter what their level are pre-exposure, are undetectable. So antigen-antibody complexes are almost immediately distributed into the reticuloendothelial system, and it's really our view now by looking at these detailed kinetics are what happens in the first week has more impact on safety and ultimately the efficacy than very low level pre-existing antibodies. And so I'll talk a little bit about these effector cells, particularly focusing on B-cell immunity because that's what is the usual exclusionary criteria. So just to say, a lot of these principles have been fabulously covered in the first and second session, but I do have a couple of summary slides to make a few points. And I raise this in the questions that we don't have an assay that currently distinguishes between seronegative individuals and one who are truly naive. These assays have been difficult to standardize because they are companion diagnostics and are developed by a sponsor for their product. So we generally don't have lots of details on the how the assays are done when patients are trying to determine their eligibility without enrolling in a study or being candidates for commercial therapy. I will say that most of our experience, at least in our group, is with the binding assay. We've developed this almost 20 years ago, have done almost 1,000 of these binding assays, and I'll show you some data on longitudinal data that Kathy mentioned, I think is very valuable to understand what we're up against in terms of antibody responses. But to one aspect of the binding assay, even though we can improve the detection methods to increase the sensitivity of these, we find that these are the most reliable at really the lower limit of detection. And that because there are matrix effects of the plasma added into this transduction inhibition assay, in some cases that actually enhance transduction. So we think that by diluting either into seronegative sera or albumin, you can avoid some of the matrix effects. And we use an external control in our TAB assay to achieve some consistency. And so what you'll see are not dilution units, but units per mil based on the external control. And what we're trying to do now is with over 50 pooled positive control sera, which are very high titer, take the top 5 antibodies that can be identified against capsid, generate monoclonals for them, and then make that available as an external control for the community to try to achieve some standardization. But here's the bad news about seropositive status. And in fact, last month was the 25th year following exposure of the first patient in a muscular dystrophy trial given an IM AAV1 vector, and he's still seropositive. So for those that are waiting for antibodies to go down, that's not likely. We did follow a cohort of patients, 113 of this larger cohort, out over 5 years. A fewer portion of them are that long after enrolling in this study, but we see that they remain very stable. And that's partly because the antigen persists in lymphoid tissue and continues to prime and boost over time. So just to kind of go into now what we've looked at as approaches to addressing seropositive status. Obviously, you'll hear a lot about the work we've done for this antibody B-cell depleting strategy with rituximab and the potentiating effects of rapamycin, not just their anti-T cell effects. There's been work done at our institution by Mavis and Rob McKenna to re-engineer AAV to reduce immunogenicity, and particularly finding specific epitopes on the threefold axis of AAV where many of these antibodies bind and prevent entry. There are other plasma cell targeting agents, ways to target IgG that you've just heard about, which are very encouraging, and ways to also deplete them or reduce through decoys, as Kathy mentioned, modifying the AAV capsid, which I think may work for the pre-existing antibodies, but once the exposure happens, all those are neoantigens anyway. So this is just to remind us that there are many of the key receptors here, TLR2, most relevant to the capsid activation. But I'd like to call attention to work that Roland Herzog's done on the role of TLR9. So this actually is the main sensor for CpG islands in the vector genome. And despite the fact that we make vectors in eukaryotic cells, they actually mostly carry the CpG signatures of the bacterial DNA that's used to make the vector. So there's not, in the current strategies used for manufacturing, not significant rounds of replication that would lose those CpG signatures. That's why there's always been some attempt to reduce the CpG islands within the expression cassettes, but then there's a trade-off because those often reduce expression. So, and I'm not going to say a lot about the pathways for anti-transgene immunity with the amount of time I have. But I do want to come back to this issue of what happens in the different compartments that we're looking at. Obviously, it's easiest to access the blood, so the fluid phase of the reaction in the vasculature is where we have the most opportunity to collect data. Many of these reactions, like the toll-like receptors, occur on the cell surface, and this is where the surface antibody matters, and wherever the vector goes, the antibodies go. And so we see that there are also then intracellular activation of what I mentioned, the impact of the vector genome itself. And not to discount the fact that the negative charge of the vectors and their activation of endothelial cells is another big part of the reaction on the surface of the blood vessel. There are interesting parts about AAV's interaction with complement that I'll get into in a minute, and I'm going to go quickly through some of that because it's been published. But the main approach we arrived at, because one of the indications we're studying is a congenital myopathy and Pompe disease, a significant portion of patients are CRIM negative. We knew that their anti-drug antibodies form against the current standard of care therapy, replacing the enzyme. And we had some history of understanding what approaches to blocking anti-drug antibodies were in that population, and some safety data in infants through school-age children. So using this same approach now to address the anti-capsid immunity, we relied on the ability of rituximab to kill B cells and pre-B cells, but it's important to note that even though an early plasmablast may have CD20, it's lost when they mature and start to make antibodies and become plasma cells. So someone that has a true memory B cell and in fact a prior exposure will not be successfully blocked from generating antibodies by rituximab alone. And so we think that the cytotoxic T cell response is influenced by the sirolimus, but we found in primate studies that sirolimus had a very important potentiating effect on the B cell population as well. And certainly allowed a defined amount of rituximab to be given. It's important to realize, though, that some of those cells are in the lymphoid tissue in the thymus, and they're not only in the circulating blood. So it may be tempting to say a single dose of rituximab depletes the peripheral blood B lymphocytes, but there are many that still reside in the spleen and other lymphatic tissue, and they will convert to plasma cells. So if the full ablative dose of rituximab is not given, you may get an attenuation, but not an elimination of the antibody response. We first studied this in 2017 in a cohort of 9 patients, 6 of which had no immune modulation, and 3 of which were on rituximab and sirolimus because of their standard of care therapy with enzyme replacement therapy. And we found that those that were maintained on rituximab and sirolimus had absolutely no anti-capsid antibody response. That was encouraging enough to then in 2019, the first patient who received systemic AAV therapy for Canavan's disease received the same regimen, and actually now 5 years after that treatment, he remains seronegative for AAV delivered systemically, and that was an AAV9 product. Just to support this, a simple experiment in mice, these 4 groups were considered a control not expressing GAA, 3 groups that expressed GAA that either did or did not receive immune suppression or had a single or a second dose. This is the result of that experiment. You can see that if a vector doesn't make GAA and you don't give immune suppression, you don't get any antibodies, so important negative control. The positive control here, the group that's not immune suppressed, has a response to the human transgene product. But then in terms of the capsid, obviously that GFP vector still produces an anti-capsid response. The 2 groups that got either single dose or double dose under this immune suppression remain seronegative, and the positive control, is responsive. So this and primate data was sufficient to approve a study that started at the beginning of the pandemic, not necessarily a great time to give people rituximab, but this study is still ongoing in 2 subjects enrolled thus far. We showed that we actually can randomize to the IM administration of one leg versus the other in a blinded fashion, and the patients got 2 doses of the intramuscular gene therapy and had expression and no response after the second exposure. There is a limited window, though. We did find that in the primates and in another study done at NIH with Cindy Tift, that after B cells are restored and there's still residual antigen present, whether that's 6 or 8 months after the induction of B cell depletion, they will seroconvert at that stage, but that's usually outside of the safety window concerns. With this data about B cell depletion, we started to learn in systemic AAV studies, done now the first one about 5 years ago, that these antibodies, especially IgM, can contribute to activation of the classical pathway. So this endothelial injury leads to platelet consumption, and then further injury of the endothelia occurs when the membrane attack complex is formed. Predominantly, this antibody-dependent pathway is through through the classical pathway. But there is also alternative pathway activation. And as we point out, unless we're looking in detail at factor B and looking for the alternative pathway specific findings, it can be hard to distinguish these because they wind up in the same place. The most effective way to address this will probably be through products that work directly on C3, because this alternative pathway can be activated in the is activated in the absence of antibodies. And it's also worsened, I should say, by the persistence of vector in the blood. So the longer the vector is exposed to C3, the direct cleavage of C3 leads to this C3 convertase that ultimately contributes to the membrane attack complex. So a very complex process, but we found that the sort of downstream inhibition of C5 convertase, either in response to TMA or preventatively, was not very effective because there's very strong pressure on C3a and C3b. And so blocking the antibody response, the most proximal point, was more effective. C1 esterase inhibitors were also not effective. So we published this paper in January, which really described how these two pathways interact in systemic exposure in a cohort of patients across a variety of studies, all clinical data with the attempt to acquire this more granular information, complement phenotyping for the first 2 weeks after exposure. Here's the way the patients were divided: 23 patients that received conventional glucocorticoid immune suppression, 15 that received this immune modulation therapy that we say beyond steroids with rituximab and sirolimus, 2 adult patients and adolescent patients in the Danon study, the largest number at this NIH study with GM1. These are mostly children in 2 dosing cohorts, and 2 SMA patients who are actually seropositive at first presentation, and then with alternative treatment for a period of time to let the antibodies decay and become eligible for commercial therapy for Zolgensma. They received immune suppression, and in fact, I will point out that one of the impacts of this reduction in antibody levels is a delay in the clearance of the product. We heard, actually, I think Juliette showed some similar data. That actually leads to a greater efficacy, between 2- and 4-fold greater level of transduction at the identical dose. Just to highlight the safety issues, and then and I'll get to what we can do about all of this. We see that endothelial injury leads to platelet consumption almost immediately. 50,000 to 100,000 decline in platelet count within a day. That can even lead to single digits. Interestingly, though, unlike ITP or other hematologic causes of thrombocytopenia, the platelets are hyperfunctional in this case, and we saw no impact on bleeding. I'll leave this to look at the paper, but this basically, I want to point out one thing. It's the kinetics of the loss of the vector and the rise of the antibody. So this intersection is what ultimately leads to unwinding or lessening of the effect. The other key element we look at is D-dimer, or the membrane attack complex. Those were eliminated in patients that received immune blockade. Same for the C5a. It's a key measure. In vitro, we can measure this across various types of vectors, whether you titrate in the antibody or titrate in different amounts of vector. Both the alternative and the classical pathway can be activated in vitro. So just to say in the end, what can we do about it? Here are a variety of mechanisms. I described the rituximab approach. We've also looked at dartumizumab in a paper with Roland Herzog, the effect of belimumab and rituximab. And then we have a group I'll show you with bortezomib. Basically, these various strategies are laid against one another. We see actually the greatest effect when you use a proteasome inhibitor as well as CD20 and sirolimus. But we also found that this idea of blocking recycling has a similar effect, a very pretty benign therapy of the Fc receptor portion of an antibody. I will add to that and I'll show how that study is being designed as I finish up, but I just wanted to say one thing about the acute antibody depletion idea. In some cases, we've need to use plasmapheresis as a rescue therapy, and this one critically ill child in a Duchenne study had to undergo plasmapheresis because of severe hemodynamic instability. So we remove the antigen-antibody complexes, and you can see what happens. They return in 24 to 48 hours because the plasma cells are not depleted. This might mean that those antibody cleaving technologies are not appropriate after exposure, for sure. Luckily, this worked. This child recovered. The downside of it is you also remove some of the product, and so impact efficacy. Vyvgar's been touched on. I won't go into this, but I'll say we've submitted an IND to the agency for studies supported by MDA, Cure Duchenne, and PPMD. Two groups, one that have pre-existing antibodies, a group that are post-exposure. We don't expect that this post-exposure group will have as much impact as those pre-exposure, but I think it's an important question to answer is what degree of depletion. And I'll just finish by saying the last thing we think will be really useful in this area is to develop an antibody-specific degrader in collaboration with Biohaven, using the trafficking of the antibodies into this acylhaloglycoprotein receptor in the liver. You can actually degrade antibodies more effectively than just preventing their recycling. So Biohaven has developed this platform in which a library of compounds can be linked to this molecule that brings the antibodies into the receptor, and they're using this for pan-IgG syndromes now. We're working with them to develop an AAV, an anti-AAV specific degrader molecule. This can be given sub-Q. The product is recycled and continues to have an effect and could be used to help lower antibodies in patients who are pre-immune. So I'll end there. We're trying to coalesce a lot of this activity into a new center at our place. So all commercial therapies are being done in this Center for Advanced Therapeutics. So I'll end there, and we'll talk more at the panel.
Yes, definitely. Thank you. So our next speaker is Barbara Sullivan from Ultragenyx, and she'll be talking about pre-treatment of cynos with prednisolone, rapamycin, and rituximab to inhibit capsid-specific humoral immune responses to high-dose AAV gene therapy.
I will wait. Oh, there you go. Yeah. Okay. Hi, everyone. It's kind of hard to go after Barry Byrne. But actually, my talk is very much linked to what he presented. I'm going to show a very detailed two non-human primate data studies today that were largely inspired by Dr. Byrne's work. And because so many people have already covered the kinetics of the innate and then the adaptive immune response, I'm just going to jump right into the data here. So I think that in this room, a lot of us know this, but I just wanted to be very direct about it, that a lot of the post-dose safety events that have observed in AAV gene therapy clinical trials have been immune-related. So some happen very fast, like the infusion-related reaction, which can happen minutes after dosing, thrombocytopenia, or a more severe form, the thrombotic microangiopathy, which can occur 1 to 3 weeks after dosing. There's a recent case at ESGCT describing mortality about 1 week after dosing. And then longer time period after dosing, hepatotoxicity or myositis. So all of these things are very, very key for us to work on and understand. So we really wanted to focus in this non-human primate study, doing a controlled study, to understand how we can best address thrombotic microangiopathy. So we took a similar approach, as Dr. Byrne just mentioned, with the hypothesis that the mechanism of thrombotic microangiopathy has to do with the overlapping kinetics of the capsid in the blood and the antibody formation. So for example, in scenario 1 on the left, if you have a low dose or a short half-life capsid, the capsid is cleared predominantly prior to the emergence of high titer anti-capsid IgMs in the blood, so assuming a seronegative patient. However, scenario number 2 on the right, with like a longer half-life capsid or a higher dose, there's still sufficient capsid in the blood as IgM starts coming up, forming immune complexes. We know from years and years of work on the complement cascade that IgMs are very, very potent at activating the complement cascade, and the more immune complexes you have, likely the higher amount of complement activation. So you might see a little bit and small amount of platelet reduction or quite a lot in thrombotic microangiopathy. So we used this as the basis for our studies, where we wanted to understand the very best way, the most efficient and effective way to prevent the formation of anti-capsid IgM as well as IgG. And so we took a similar approach. We were using a high-dose AAV gene therapy at 2e14 vector genomes per kilogram dose, so we intended for Duchenne muscular dystrophy. So we knew that those patients were already on prednisolone or steroids. So we included a steroid in our study. But to target the B cells, we wanted to look at rituximab, which is a monoclonal antibody that binds CD20 and depletes many B cells, but not the plasmablast or the plasma cell population. And in fact, in the Genentech studies, they demonstrated that RA patients who are non-responders to rituximab have a very high plasma cell gene signature in their blood. So we knew that rituximab would do a good job at depleting a lot of the B cells, but not enough of the B cells. So we also went with rapamycin, also known as sirolimus. And we had shown previously in non-human primate studies that rapamycin leads to a very strong and rapid reduction in the plasma cell gene signature in the blood, and also in vitro blocks the differentiation of B cells into plasma cells. So we chose this combination to most efficiently target the entire range of B cells that could be possible. So this is from Dr. Byrne's paper that was published earlier this year, where he demonstrated within this critical window after dosing, which I'm calling the TMA window of 1 to 21 days, you see a significant reduction in anti-capsid IgM on the left, anti-capsid IgG in the middle, and then a reduction in soluble C5b9, which I think is a very good biomarker for complement activity. So we had 2 studies. We're trying to figure out what is the most efficient and effective. Could we just use rapamycin and steroids? Could we just use rituximab in order to reduce the patient burden? So the study on the left, there were 5 groups. We had a control arm with no vector, no immunosuppression. The pink group is the immunomodulation group only. Then we had a steroid only, steroids and rapamycin. And then the triple immunosuppression down at the bottom, there's prednisolone, rapamycin, and rituximab, which I've started calling PRR, which my innate immune colleagues don't like, but I can't help it. So we discovered in study number 1 that this combination, the PRR combination, was the most effective. So study number 2, which most of the data I'll show you today is from study 2, we wanted to know whether rituximab alone could be sufficient. Oh, and so a couple more things on the slide, sorry. The vector dose was 2e14 vector genomes per kilogram administered intravenously. Our doses were selected for the steroids. This is 1 mg per kg per day. It's a typo, it's not 1 mg per gram per day. 1 mg per kg per day, which is a very standard human clinical dose. The rapamycin, in our first study, we did a therapeutic drug monitoring aiming for 12 to 20 nanograms per mil in whole blood. But we found that actually the animals that had the higher exposure had the better response. So in study 2, we did a fixed dose of 2 mg per kg per day. This is the cancer dose, and this was for monkeys. And so I don't think this is necessarily the right dose for humans, but we were looking at pharmacodynamic effects, and we were trying to use the PD of the immunosuppressants to achieve our results. So a human dose would probably not look like this. Likewise, for rituximab, we chose our dose of 10 mg per kg per week based on Genentech preclinical data, showing that this could very effectively deplete B cells in the peripheral blood. Again, that's not a human dose. This was a cyno dose because we're using cynos. The other thing I think I'll say about this study design slide, so you'll see that we start on study day 1, we administer our vector on study day 29. Okay, so we monitored the rapamycin drug levels. Study 2 is shown here. The trough levels were between 10 and 35 nanograms per mil in whole blood. It was well tolerated. In study 1, we did have 1 animal who had a severe weight loss, so we tapered them off the rapamycin. The rituximab, we did see a very efficient B-cell depletion. In study 1, we saw 100% depletion in the PRR group. In study 2, because we had, which I'm showing here, on the pred-retux group only, we saw between 75% and 100% depletion in the peripheral blood. And in the triple immunosuppression or PRR group, we saw between 97.8 and 100% reduction. So indeed, we saw better reduction in the peripheral blood and the B cells. When we compared the anti-capsid IgM antibodies, the PRR group had a lower antibody response over time as compared to the no immunosuppression group. The anti-capsid IgG antibodies, we saw a similar finding that there were lower antibodies in the PRR group, and I've really highlighted the first 21 days after dosing, which is the TMA window. And then summarizing, looking at the last collection of both studies, so study 1 was a little bit longer at 113 days, study 2 was 50 days. We saw a reduction. We didn't perform any statistics due to the very small sample size, but you can see this is a log scale, and the PRR group in both cases, which is the one on the very end, was a lot lower than the one on the left. So then we were interested in, well, how does the B-cell depletion in the peripheral blood relate to the antibody titers? And what we found was that if you look on the x-axis on the graphs on the bottom, percent peripheral blood B-cell depletion on study day 29, which was our AAV vector dosing day, versus antibody titers on day 50, it's very, very flat, and then there's a sudden cliff, right? So you have to actually get very, 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 very close to 100% reduction in the peripheral blood to see any impact on the antibodies. And that's summarized in the table on the top, where the animals that are highlighted in green had 100% reduction or 99.1% reduction, and either on the right, negative or 1 in 20 titer antibodies. And then on the left, even though they had 100% depletion, they still had detectable titers, but a lot lower than the rest of the group. So we really feel that near complete depletion in the peripheral blood prior to AAV gene therapy dosing was required to see a clear impact on the anti-capsid antibodies, whether it's anti-capsid IgM or IgG. So then we looked at various tissues. So what is the impact of the prednisolone, rapamycin, rituximab pretreatment in the serum? 4 days after dosing, we saw more than a 1,000-fold increase in vector genomes. We were pretty excited to see this. However, when we looked at various tissues of interest, we did not see a very, very large increase. In the liver, we saw about 2-fold increase. The quadriceps made a 3 to 5-fold increase, but other tissues were more variable, including the diaphragm and other key muscles for Duchenne muscular dystrophy. So it was kind of inconsistent, even though the serum effect was pretty striking. Regarding the complement, we also, like other folks have shown, we saw an increase in soluble C5b9 4 days after dosing, and BB 4 days after dosing in the control in all 3 groups in study 2. Interestingly, in both study 1 and study 2, all the animals that received rapamycin, we saw a steady increase in complement that increased over time. We also saw a couple other acute phase reactants that sort of steadily increased over time. So I think this is an important consideration for the chronic use of rapamycin. Not shown here for time, we did see a transient platelet reduction also 4 days after dosing in the animals that had the complement increase. So to conclude, the gene therapy at 2e14 vector genomes per kilogram IV and the immunomodulation combinations were generally well tolerated in the cynomolgus macaques. The combined treatment of prednisolone, rituximab, and rapamycin was the most efficient and effective combination to reduce anti-capsid IgG and IgM within the first 2 weeks after dosing. In a post-hoc analysis, we analyzed that near complete depletion of peripheral blood B cells prior to gene therapy dosing was required to see clear impact on anti-capsid IgG and IgM. In our biochemical analyses, we demonstrated complement soluble C5b9 and BB increase in all groups, not all animals, but all groups, 4 days after dosing. And there was an unexpected persistent upward trend in all rapamycin groups. Platelet reduction was observed in several animals from all groups, again, 4 days after dosing, and this resolved. And the prednisolone, rapamycin, rituximab groups had higher serum viral genome copies 4 days after dosing, but inconsistent improvements in transduced tissues. So with that, I'd like to thank my colleagues. Denise, Leah, and Lorelei are in the audience. They're waving. There are a lot of almost all Ultragenyx contributors here on the left. Brian McIntosh is from LabCorp. This is a cartoon of our brand new laboratory building in Somerville, close by, and I was there yesterday. It's beautiful. And we also have both labs and offices in Somerville and in the San Francisco Bay Area. And these folks and I all form a group which is called Ultra Immunology, and we're slaying the dragon of immunology. And our goal is to pioneer innovative and adaptable immunologic solutions that complement rare disease therapies. So, thank you.
Thank you, Barbara. So we'll round out the session today with my colleague and collaborator at Regeneron, Nick Giovannone. And he's going to tell you a little bit about what we've been doing at Regeneron with immunomodulation strategies for overcoming antibody responses to AAV.
Okay. Hey, while we wait for the slides to come up, I just want to say thank you so much to Michelle, to PPMD for organizing such a great conference and putting together such a great program and inviting us to speak and share our work that we're doing at Regeneron. So as Andre said, I'm Nick Giovannone. I lead a team at Regeneron, the newly formed Regeneron Genetic Medicine. And the focus of my team's work in conjunction with Andre is developing new immunomodulation strategies to overcome some of the challenges of AAV gene therapy. So I think this probably needs no introduction at this point of the conference, but I'll just say that just highlight the fact that there's really two distinct but related challenges. One is, of course, in seronegative patients, the development of antibodies that prevent redosing, which is a huge challenge, and then the existence of pre-existing antibodies from natural exposure to AAV. And just to highlight again that these tend to be very high-titer, cross-reactive, and long-lived. And these pre-existing antibodies can be on the lower side, and it looks like maybe long-lived too. But how we deal with these in terms of immunomodulation, actually the strategies that we probably need to take are quite different because the goal is different. So in the case of redosing, we're trying to keep AAV-naive individuals naive. And so that carries with it some different implications in terms of which cell types we need to target, as Barry and others have talked about. But in the case of pre-existing immunity, we think these responses are, to a large part, coming from plasma cells, and that means that we have to approach immunomodulation differently for that, too. So at Regeneron, we're a company that specializes in development of immunomodulatory drugs, so we thought we're kind of in a good position to tackle this question and maybe even apply some of our late-stage clinical therapeutics to these challenges to make a more immediate impact. So in the first part of my talk, I'll address how we're approaching the redosing question, how to keep AAV-naive individuals naive, and then in the second half, I'll talk about the challenges of pre-existing immunity and how we're dealing with that. So in thinking about the redosing question, it's helpful to think about the B cell response to AAV in two different parts. If you have a naive B cell that encounters AAV, it becomes activated through interactions with T cells. Those interactions lead to the development of what's called a germinal center. And these germinal center responses, there's continued interaction between B and T cells that lead to B cell proliferation. Importantly, also differentiation to long-lived immunological memory, as well as antibody affinity maturation. But once you get to this stage where you have long-lived immunological memory, the strategies that we have to take to pursue this is a little bit different than preventing the response. And so in thinking about how we can prevent this response, one of the receptors that is really critical to this whole initial process of activation and germinal center response is this TNF family, superfamily receptor called CD40. It's critical for interactions with T cells through CD40 ligand, CD40 interactions. CD40 is on the B cell. It's also actually important for promoting T cell activation via DC licensing. And then for the germinal center response, it's a really central receptor, both in promoting antibody affinity maturation and then importantly the development of long-lived immunological memory. And this receptor is known to the AAV field. It's been, the ligand has been targeted before by some of you in the audience actually. But we at Regeneron have some antibodies that target the CD40 receptor itself. And these are fully human effectorless IgG antibodies. And we wanted to apply these antibodies to this challenge and see if we can use transiently blockade CD40 to suppress the anti-AAV antibody response and importantly prevent the formation of B-cell immunological memory. And in doing so, keep naive individuals naive and enable redosing. So we have a variety of useful tools at Regeneron. One area that we're really good at is making humanized mice. And so we have these CD40 humanized mice. And we tested these two anti-CD40 antibodies that we have that target human CD40. We prophylactically dose these mice 25 mg per kg with anti-CD40 and then twice per week for the first couple weeks. Then we, at day 0 here, we dose with an AAV8 vector and then analyze the total antibody binding titers in these animals. And so what you can see in purple and orange are the CD40 antibody treated animals. You can see we had really strong suppression of the anti-AAV titer response by about 3 months after treatment. These titers had returned to what we see for the range for naive animals, and we were able to, we dosed these mice with a second AAV8 vector GFP. And when we looked at transduction of the GFP vector, which was dosed at 6.6 E12 mg per kg, you can see that the CD40 antibody-treated animals showed similar levels of transduction for GFP DNA, RNA, and protein as what we saw for animals that had never seen AAV before and only got the second vector. And the GFP immunohistochemistry you can see here, very robust amounts of GFP staining quantified here on the right. So we were excited by these results, and we wanted to confirm the MOA of this approach. I mean, as I said, CD40 is really important for the germinal center reaction, which is critical for the B-cell differentiation to long-lived immunological memory. And when we looked at AAV-specific germinal centers, we found that they were almost entirely suppressed, completely undetectable, actually. But that's consistent with the MOA of CD40 blockade. And then for total germinal centers as well, they were also completely suppressed, suggesting that CD40 blockade, consistent with what's seen in the literature, could prevent germinal center formation, new germinal centers, and also ablate ongoing responses as well. And then I briefly mentioned that CD40 ligand interactions are important for the T-cell response to AAV as well. We know that from Roland Herzog's lab's work. And so we looked at the AAV8 capsid response by interferon gamma ELISpot and found that these antibodies also were able to suppress the anti-AAV T-cell response as well. So the hope is that through CD40 blockade, which has kind of had a renaissance in recent days, that this approach could be really useful for preventing both the B and T-cell response to AAV and enable redosing. Okay, so what about this other challenge, the seropositive challenge? What do we do in that case? So I'll highlight here that what we think are the really important cells are the plasma cells. And these are the terminally differentiated B cells. As Dr. Byrne mentioned, they tend to downregulate many features that make them B cells. So CD20 is downregulated on these plasma cells. They're highly prolific antibody-secreting cells. They can hang out in bone marrow for literally decades. And as I said, they're the source of most circulating antibodies. So in thinking about approaches to target these plasma cells, which are the likely reservoir of long-lived AAV immunity, CD20 antibodies, they deplete a lot of B cells, but they don't really touch plasma cells. And we heard some really promising approaches with IgG degradation, but you have the added challenge with that you still have plasma cells hanging around that are continually pumping out the antibodies. So you can get rid of the antibodies that are already there, but some amount of antibody is still going to remain because the plasma cells are pumping out those antibodies. So we wanted to test a strategy of plasma cell depletion, and at Regeneron, we have BCMA by CD3 bispecific antibodies that can quite effectively kill plasma cells. And so mechanistically, how do these antibodies work? As I said, they're bispecific antibodies. They bind CD3, which is expressed by T cells. They bind B-cell maturation antigen, which is highly expressed, uniquely expressed by plasma cells and late-stage plasmablasts. And what it does is it brings those two cells together and induces cytotoxic killing of plasma cells. So as I said, plasma cells, as they differentiate from B cells, they lose expression of CD20 and eventually CD19 as well. And as they're losing that, they're gaining expression of BCMA. So this approach, as you know, Andre's team has shown that this is quite effective at targeting plasma cells. This here on the right is showing bone marrow plasma cells, and you could see at different doses of BCMA by CD3, you get quite effective depletion of the plasma cell reservoir in the bone marrow. So the question was, can we apply this strategy for eliminating pre-existing antibodies to AAV, the plasma cells that are pumping out these pre-existing antibodies to enable AAV gene therapy? And so again, at Regeneron, like I said, we're great at making humanized mice. So we have BCMA humanized mice. We have CD3 humanized mice. So we were able to use BCMA and by CD3 that targets both the human versions of these proteins. But one thing we don't have is mice that have pre-existing AAV antibodies naturally. So we had to treat these mice with an AAV vector to induce pre-existing antibodies. These, of course, when you do that, you generate really high titer responses in mice. So we did that, but then we waited about 2.5 months to let the primary response mostly subside and have the response mostly be contributed, what we think, by plasma cells. Then at that point, we started them on immunosuppression. So we had a group in yellow that was treated with BCMA by CD3 to deplete the plasma cells. Then as comparisons, we also included anti-CD20 and anti-CD19 antibody-treated mice to deplete the B cells. We also included efgartigimod-treated animals to compare versus targeting the IgGs through FcRn blockade, as we've heard. But when we did this, and so these mice got this weekly for several weeks, but when we did this, we didn't really see a great impact on the pre-existing antibody titers. So this was a little bit surprising that we saw nothing. But fortunately, in this experiment, we also had the idea to combine the different treatments with the rationale that BCMA by CD3, it's targeting the plasma cells, but the IgG antibodies that are already around are hanging around for quite a long time, probably because of the action of neonatal Fc receptor. So we thought if we were killing the plasma cells, maybe we were getting rid of the production of IgG, but the antibodies that are there are still there and hanging around. So when we combined the two together, BCMA by CD3 with FcRn blockade, with the rationale that we get rid of the source of the antibodies and then turn over the antibodies that are already there even faster, then that's when we start to see a much more potent effect on the antibody, the pre-existing antibody response. But you can see even with this combination, we didn't quite reach, in some animals we actually do reach baseline, but on average we kind of saw this plateauing out effect with these two together. And so we also had animals that were treated with all three immunomodulators. And when we did that, we were able to bring animals down completely to LOG. In all 6 mice here, they had undetectable antibody titers. So the question is, why do we think we need all three? And I think there's a couple possibilities, but one that's stronger than the other. One possibility is that we treated these animals with AAV. We think the primary response mostly subsides 2.5 months later, but as we know, these capsids hang around a long time. So it's possible in this mouse model of pre-existing immunity, there's still a primary B-cell response that's happening that B-cell depletion can address. And maybe that's part of the reason. But what we found actually is that efgartigimod, which is a human IgG fragment, is quite immunogenic in mice. So after a couple of doses, you start to completely lose efficacy, at least in our mouse system, due to anti-drug antibodies against efgartigimod. And those anti-drug antibodies happen to cross-react with BCMA by CD3. So it's the whole point that I'm trying to make is that we don't actually yet know what combination is required in in species other than mice, but in mice we see that the triple combination works most effectively, probably by reducing immunogenicity of efgartigimod. Okay, so what about the AAV redosing now at this point? What do we see? Here I'm showing GFP immunohistochemistry, which was the AAV8 vector that we used. You can see that BCMA by CD3 alone was not effective on its own, but when we start combining it with efgartigimod for FcRn blockade or the triple combo, you can see that we have control mice starting to look a lot like the triple combination treated mice. And that's quantified here on the right. And when we looked at DNA and RNA of the GFP vector, you can see that the double combination, there's some mice that achieve transduction at levels of controls, but the triple combination is most effective in this system. Okay, so to try to summarize this in a very simple schematic, oversimplified schematic, if we think about AAV antibodies as kind of like water in a sink, you have the plasma cells that are acting like the faucet and contributing these antibodies into this water reservoir. And then you have some sort of antibody clearance rate, that's your drain. And so we tried, depleting the plasma cells, and that turns off the faucet, but maybe there's still some antibodies that remain that aren't quite draining out. So and this, like I said, is probably due to the action of FcRn, which keeps the IgGs around. But then when we combine both plasma cell depletion to turn off the faucet and then accelerate IgG clearance with FcRn blockade to widen the drain, now the sink is depleted of all those antibodies. So I'm going to end there because I'm right at time, but I'm happy to take questions during the panel discussion and afterwards. Thank you very much.
Okay, so I think we're ready for the speakers to come back on stage, please. And we're also going to invite Sherry Gordon and Damon Asher from AskBio and Sarepta to join us.
Michelle said 3:30. 3:30.
So I guess I can kick us off, and I'll start with the first question that I thought of during the session, which was for Carsten, which is you mentioned this T-cell cytotoxicity that you tried with your immunomodulation therapies, but we also heard from Nick that B cells can have an antigen presentation impact, and in particular in the CNS, that's been seen, for example, in cases of multiple sclerosis. So have you thought about these B cell immunomodulation therapies?
It's true, but it's also true that rapamycin that we chose is not just T cell selective. It really has an effect on the B cells as well. That was shown in some of the diagrams here as well. And so I think we are in our results that I showed in the last slide, that actually this regimen also has an effect on the neutralizing antibody response against the capsid in the end. Tells us that we're addressing both axes. The preclinical data was mostly T cell toxicity that was seen. Steve Gray found by delivering admittedly quite immunogenic foreign protein. I think he delivered GFP to these. And so that was a really T-cell heavy perivenular kind of encephalitis that was managed with or prevented with rapamycin. So you're right, we're probably seeing hopefully both axes being engaged. I ran out of time, but implied in my last slide on the ELISpot was also that we never saw any ELISpot response against the transgene under that regimen. We don't have a good antibody assay against GAN, so we can't measure anti-drug antibodies in our system.
I have a question and consideration. The question is for Nicolas, right? I noticed that in your re-administration experiments, and I don't know in the second where you do the combination. Here's the light. I noticed that the second dose was actually a lot higher than the prior dose, right? And in a way, the question for you specific is whether you tested the same dose, and the consideration for the panel is that we hear a lot about strategies to lower antibodies following the first administration and then to knock them down further. But then we heard that there's a dose consideration. So it seems like that in a re-administration setting, one thing to keep in mind is that we may need higher doses to kind of overcome that little bit that stays around.
Yeah, no, I think that's an excellent point. And we have tried higher doses, and we do see there is a higher dose of AAV, of course, leads to stronger antibody response. And we've kind of seen mixed results with the CD40 blockade. You know, in some cases, it's quite effective at suppressing even at high doses. And then in other experiments, it's less effective. So we're still kind of sorting that out. I think one thing to consider is CD40 blockade primarily impacts a T cell dependent response. And AAV capsids they have repeating subunits that can favor a T cell independent response. And so it's possible that we might need a combination of approaches that addresses both of those types of B cell responses. But like I said, in some cases, even at very high doses, we've seen suppression with CD40 as a monotherapy. So it's still under investigation, but it's a really good point.
Thank you. I have two questions. Firstly, I just heard about a lot of wonderful discussions using anti-CD40, CD3 combinations for B cell and even degradation using PROTEC likely methods for degrading some molecules. So my question is that it's possible we can use RNA medicine like transiently modulate cell-cell communications to modulate macrophage and APC, dendritic cells, to modulate the immune response transiently, so that to open a window for AAV-based therapeutics.
So you're talking about like siRNA type approaches, or
Yeah.
I don't know. I think it's a really interesting idea. I think it's certainly possible. I don't have any data to speak of on our end, but I think that's a really interesting idea. Yeah. No, I don't know if anybody has any comments on that as well, but it's certainly a great transient suppression idea. Yeah.
So, I'm going to add one thing that relates in a way to Fed's point. In many cases, we're thinking of what would be the opportunity for remote redosing that may be years later after there's a diminution of the primary effect. I mean, this has not been necessary in neurons or cardiomyocytes or truly terminally differentiated. I think the biggest issue is in the musculature. But there are also possibly opportunities for divided dosing in a near-term period, because I think we're often saturating many of the cellular receptors on first exposure. So I think we have to differentiate near-term, intermediate opportunities for redosing versus long-term remote redosing strategies. They may involve different aspects. The one thing that appeals to me about this intermediate dosing interval idea is that in lieu of having delayed treatment cohorts in a study, you could assess the acute toxicities in a within-subject dose escalation paradigm, which is blinded. And so in a much smaller study, you'd both assess safety and the efficacious dose, and so that no patient would ever be exposed to the ineffective dose or minimally effective dose. And that could be done in roughly a 3-month time window. I think that would be interesting if we can get that idea socialized with the agency.
Yeah, I would completely agree with that, and I would say that part of the reason that we're so excited about the data that Nick showed with CD40 is because it might enable you to do
Create
that
that window.
and prevent the nascent anti-AV response and really allow you to build up and learn as you go about dosing and about
I love the kitchen sink analogy. I think one of the reasons we're having to pile on all these things, including the kitchen sink, is that cells are constantly undergoing maturation. So it's hard to get a protection window long enough when there's really your immune modulation is around, the cells are amenable to being blocked, and the capsid's gone. You know, that is a moving target, and there are always cells that are not going to have the ligands that you're hoping to target with the therapeutic. So it does make this remote redosing a little more challenging than this window. Even a 6-month window is very achievable.
Yeah, and just to mention, I forgot to mention to Federico's question earlier, with the CD40 blockade, one of the key things is that we're preventing memory formation, we think. So that would allow for these longer-term approaches. So specifically related to that, you know, if we can prevent the memory, then hopefully sometime down the road, if it's 6 months or whatever, then yeah, we can redose. Even if the initial response, there is an initial antibody response, if that subsides, then we can redose.
It certainly, once the long-lived plasma cells are a substantial population, they're never going to go away.
I had a question related to that, Perry. Is that okay?
Yeah, sure.
You look like
Go ahead.
yeah,
No,
not
I was
really.
just signaling. Go ahead.
Okay, so do you have to cover CD40 or 40 ligand for a whole year? Like, what happens when you come off CD40? So Lily Wang has this lovely paper from 2011 where she shows that follicular dendritic cells are still holding onto the capsid many, many months after dosing. And in mice, you look up and like they say, we tested for a year and then we stopped because it's still there. Right? People just stop because follicular dendritic cells are so good at holding onto things. So what happens when you take off the CD40 blockade?
It's such a good question. It's such a good
You
question.
haven't done the experiment.
We haven't done that experiment yet.
Well, because potentially that means you have to have this IV treatment every 2 weeks for a really long time in order to enable the second dose.
Let me just say about the dosing. We that was like a maximal dosing experiment just to demonstrate POC, but we don't think that we need twice a week dosing. So I don't think, in terms of what would the relevant human dose be, we don't know yet. Hopefully it would be a lot less, but we don't
I
know.
think these are the questions that we'll have to sort out following the POC experiments that we've
Yeah.
done. And I think that figuring out the dosing, the duration of the dosing, and exactly your question, what happens if there's still capsid around when you take off the CD40, all of those are going to be
I mean,
critical
MedImmune
questions.
had a CD40 for a long, they looked pretty good for a while, which also,
There have been a lot of companies developing,
Yeah.
and they've
yeah, various
Some of
stages of
them-
development.
some of them are
Okay.
still around,
So
some of them.
We had one observation in that expanded access study for Canavan's disease. It was not by design because the intended window of coverage was 6 months. But then we saw that there was persistence of vector genomes in the blood much longer than expected, because in the preclinical studies, that's generally not the case. You know, by 1 month, they're declining and almost undetectable by 6 months. But there was a very strong signal at 6 months. So we elected to give a maintenance dose of rituximab. At that point, the young man was going back to daycare, and we didn't want to have to deal with the potential for other viral exposures, so he began on maintenance IVIG. Once IVIG was instituted, which is now passively immunizing him against AAV, the vector in the blood cleared completely. And at 1 year, we stopped the IVIG. B cells had been restored by then, and he remained seronegative to this day, which is now 5 years later. So I don't know that that's a practical regimen, but it does tell me something about the influence of the residual antigen that persists for such a long time.
I guess to follow up on that topic a little bit, I just wanted to ask a couple questions about.
I don't think they can hear you online.
There
There you
goes.
go. There you
There
go.
we go.
Yeah.
Just related to TMA and thrombocytopenia. So it sounds like what I'm hearing is that the persistence of capsid and the upregulation of antibodies is really key. And is this made worse in patients who are actually naive versus seronegative? So are naive patients, because they're coming up with IgM, going to have a worse response? And just another follow-up question, is AAV9, because of its long persistent half-life, more of a risk than other capsids?
So several people alluded to this. We're hoping to, we haven't completed this analysis yet, but either with commercial therapy or in various studies that we're doing, since we're getting these daily time points, we're studying not only the antibody and complement profile, but the vector genome biodistribution. So I think it does have something to do with where those two lines intersect, because at least RH74, which has a seemingly more favorable safety profile, probably clears faster. Maybe targeted capsids will clear faster, ones that have a specific ligand. So I think those two factors and the influence of the direct interaction of the capsid with C3 also appears to be different among serotypes. And we haven't figured out that mechanism yet, but I think those two things, the clearance and the alternative pathway, are contributors, because once the alternative pathway is kicked in and you've got antibodies, there's an amplification effect. And that plus whatever product characteristics, critical quality attributes of the product might activate TLR9. These are all kind of harder things to measure, but I think you know, the magnitude and the rate of antibody response is the consequence of all of those factors.
So I'm really intrigued by the IVIG, what you just mentioned. I'm curious, then, could IVIG be with a targeted capsid that could be delivered and have an activity pretty quickly? Could really
You could, right,
follow up IVIG like
one
a day
dose
later
might be
really
sufficient to kind of clean up
To
all
wipe
the
out
residual material. Yeah.
IVIG does have neutralizing antibodies against AAV, so you'd want to screen your lot.
No, he wants it. He wants it.
That's why you want it.
Yeah, he wants for that
okay.
purpose.
I was specifically bringing up the point, if you had an AAV that could be highly effective within a
In
few
a
hours
shorter
to a
window.
day, and a day
okay.
later you come in with IVIG, could you get rid of
Yeah,
the
or a week,
business?
whatever, yeah.
So I had a question for Nick, and it harkens back to something in the first panel and the point that Roberto was making about it can be we don't really have a way to tell a true seronegative patient from somebody who's testing NAb negative. So do you think that the anti-CD40 ligand would be helpful in the case of somebody who's actually previously been exposed but is testing negative?
That's a really excellent question. We haven't looked at whether CD40 blockade inhibits memory responses. There's a chance that it might not, but we haven't explicitly looked at that. I would actually, I hesitate to predict, but, I think that's something we need to look at, yeah.
I have a question for Nick. So for your BCMA bispecific, how long is the depletion of the plasma cells? And what's that duration, and how do you think that would impact?
So it depends on the dose. We've done a much longer term study where basically they stay depleted as long as there's antibody on board. I think the inclusion of an FcRn blocker, it actually turns over the drug too, because it's an antibody drug. So, there might be some beneficial effect there where you're actually reducing the amount of time the plasma cells remain depleted. But yeah, we see reconstitution of the plasma cell pool once the drug is gone. Yeah.
Great. And I think this question I have is Barbara. I didn't quite get to the point where you show that methylpred plus retox group didn't show a higher B cell depletion, but with rapamycin on board, you saw a greater B cell depletion. What is the mechanism there, do you think?
That was kind of the point of the study, right?
Right.
What would we observe? So we didn't do lymph node biopsies or spleens or anything to really get at, what we know about rituximab from lupus nephritis patients, that you really have to have very, very deep depletion to see efficacy. But we did know that rapamycin would help on the plasma cell front, on this differentiation step between a germinal center, where a B cell enters the germinal center and wants to become a plasma cell. So rapamycin will block that step. So that's why we think that the rapamycin or sirolimus is really helping. And it just turned out to be a nice coincidence that our blood test to count B cells correlated with the antibodies, which I feel like are both very actionable, useful tests that someone could use.
It really, the regimen does matter.
Yeah.
And I think there's a temptation to try to operationalize that what's easiest would be to use one dose of rituximab, and because that
Yes.
is
No,
fast,
you don't want to do that.
But that's not going to work, unfortunately, because there are these resident B cells that are not impacted. So we found, other than what's the usual, at least for rheumatoid arthritis induction protocol, we can take that 1,500 milligram total dose and divide it in 3. And you can do that with just within 2 weeks, minus 2 weeks, minus 1 week, and right before dosing, because this is catching all of those emerging pre-B cells that are continuing to mature while this pre-treatment's going on. So it's really hard to do that in a really compact time frame, but that's the shortest regimen we were able to come up with.
And that would deplete the B cells up to 6 months in that regimen?
It depends, but at least 4 months. Yeah, so some patients repopulate sooner.
Yeah, I will say, and this is coming mostly from our experience looking at rituximab in preclinical studies, is that what happens with rituximab in circulation can be very different than what's happening in the tissue. And I think that in humans, there's biopsy studies showing that the B cells in secondary lymphoid organs are much more resistant to depletion with rituximab. So there may be a reservoir of B cells ready to replenish everything in rituximab-treated patients.
Thank you.
Just piggybacking on that question, has anybody here tried a CD19 inhibitor that would target those resident B cells?
So
It's got to be someone in human, because it doesn't cross-react with non-human primates, the inebilizumab. Have you tried it?
Not yet.
yeah, not yet, but it is something that is on our radar.
I have a question. I have a question from your talk, Barry. You had mentioned that it would be really wonderful if we could have a universal test for antibodies against AAV, all AAV gene therapies. So I wondered, just for the panel, if we wanted to create a universal test, what would that look like? And who would do it? Would it be a company? Would it be academia? What's your vision for that? I thought that was a really clever idea.
We do like the idea of an external control, whether, that doesn't preclude any sponsor developing a companion diagnostic for their product, whichever detection method they're choosing. But it becomes very hard to compare across these studies without that. And so what we started with for that purpose is a pooled positive control sera, which has got this titer of 1 to 1 million. So it becomes it's almost a lifelong supply of positive control antibody, particularly at the low range. And we continue, when patients come back for long-term follow-up, to save any laboratory tests they have for adding to that pool. So, but that's not easy to distribute widely, because the more you start sending it out, then it's not a lifelong supply. So we want to take the top frequency antibodies from that pool and make monoclonals from them.
Just do reverse engineering.
And then do a pool of monoclonals that could be distributed with an indefinite supply. So that was our idea of how to approach that, and then people can use it however they see fit. And it's not a huge cost to that, honestly.
Like a universal positive control.
Yeah.
That any lab
an
out there
external
would
standard,
want to.
exactly.
How
That's
would
fixed across all AAV serotypes.
Yeah.
Okay, that was my question. Is
Yeah.
how would you?
They're common epitopes. They're predominantly easy to find, actually.
Yeah, and if I could just comment on that, it's something that certainly is like a holy grail to be able to do a single test and have some idea of universal. What complicates that is the definition of seropositivity as pertains to, does it preclude you from getting the gene therapy? That can be very program specific, for example. And it was interesting, for example, Sarepta uses a RH74.
Right.
And in the trials we've done, by the cutoff that's set, which is you need to be less than 1 to 400, has been kind of the clinically validated value for that, we get about a 15% seropositivity rate. But it's so dependent on how you test. I mean, we saw a couple other assessments of that. It goes from like 20, like as high as 40, 60%, depending on how you test.
Yeah, it totally depends on how the dilutions are done.
Right, exactly, and also the methodology you look at. But it'd be hard to know with a universal test what is the relevance of that to actual eligibility for specific gene therapy programs, which kind of need to be validated using the tests that were used in the course of the clinical trials.
But if we started right now, we had a universal test.
Yeah.
Barry's running it for all of us. We all send our samples to him. You get the antibody for you. Sorry, signed you up. For all the things. Right. And then you do a mouse experiment like we saw this morning where you transfer in different levels and you compare that to your test.
Potentially.
Could that
or it can be used
can you use the mouse? Is the mouse good enough to be able to assess that, or you have to start like?
Well, even if it's being done in conjunction with a primate study, you just need to calibrate your companion diagnostic against the external control. That's what we've done for every patient that we have tested through a product-specific companion diagnostic. We use this. The other thing to say about an external control-based assay is the antibody level is a continuous variable. It's not categorical. So when you have someone that's on, the reason there's variability is when someone falls on a dilution, they can be eligible or ineligible by a minuscule amount of antibody. And that's what's really essentially unfair to patients that get excluded by being on the fence, so to speak. But when you have a continuous variable, it's much easier to define your cutoff because it'll be pretty precise each time.
Right, and with a continuous variable, you could imagine sort of clearly low bins, clearly high bins.
And that's really what happens. Most people are in the low bin. It's really a very small portion of the general population when you use a continuous variable that have what would be likely inhibitory to clinical benefit. And I think one thing that went into deciding what's acceptable thresholds, it was not clear in the beginning what was an impact on safety, where there might be an infusion reaction, and what was it going to impact efficacy. And I know Jerry Mandel shared with me a lot of the communications for the SMA studies where they were, where we wound up was 1 to 50. He started at a much higher level, its proposal to the agency, and there was like a negotiation, not really based on primate data or any other data. It was just like what is the lower limit of detection and what's, at least in a thought experiment would be safe, because it was essentially no antibody. 1 to 50 is very close to the lower limit of detection of most assays.
Completely different topic. I wanted to ask Barbara, you made an observation about complement levels rising in your study with sirolimus. Do you have a mechanistic explanation for that? And is it something that's been described in other settings?
So we saw a persistent increase in complement prior to dosing, also fibrinogen, D-dimer, LDH. They all generally fall into this acute phase reactant, but they're not acute because it's the whole time. There's an AAV effect, but honestly, it's just like they continuously go up. That has been described in people who've received rapamycin. We don't have a really good explanation other than potential recrudescence of viruses that are being subtly reactivated. But we don't really have a good rationale for that. But I think it is something to keep in mind if you want to use rapamycin for a long time, that there could be.
The level you had was like in the 10 range, right?
Soluble C5b9.
No, of the rapa levels. I think you said 10.
So in our monkey study, which of course is going to be higher, the range of trough levels were 10 to 35 nanograms per mil. So that's quite a bit higher than the 4 to 7 that we would use in humans. Yeah. But we found that in monkeys, we needed that higher dose level to see the effects. But humans who have received that higher dose level see this effect. Say if you have a biomarker that's an acute phase reactant, you probably don't want to use rapamycin.
We use around 8.
Yeah.
8 nanograms per mil in whole blood, yeah. We tried that. It did not work.
But we haven't tracked complement. I mean, some of our patients are now on for 3-4 years.
Yeah.
Maybe we should look.
I had a question for you.
Yeah.
Can I keep going? You saw a pleocytosis. Did you characterize what the cells were?
It mostly B cells.
Really?
Yeah.
In the CSF.
In the CSF.
In the
yeah.
CSF.
Yeah, mostly B cells. But we don't know whether they're antigen, producing B cells.
Right.
Yeah.
Did you analyze a proteomic? You saw proteins too, right?
So proteins too, a little bit of oligoclonal band activation as well. So there's clearly an intrathecal, probably we think it's capsid driven, mostly B cell predominant, that's steroid responsive and doesn't have a clinical phenotype.
Do you think it's generated locally in the brain or more in a peripheral tissue and traffics to the CSF?
I have a feeling it's probably local.
Really?
Yeah. But we don't have enough data to kind of clearly say that. And the same is true for the neutralizing antibodies that we see in the CSF. We think they're probably not just systemic antibodies showing up in the CSF. There may be some kind of local production, which makes redosing, of course, a challenge into the CSF.
Yeah.
But that remains to be clarified. We don't know for sure.
So Barry, I actually had a question for you. So for the VivGuard study, or I guess this is also true for Giuseppe in some ways, are you going incrementally, where you're starting with individuals with lower titer and then pushing it?
So in the
how high will you
natural
go?
In the pre-existing antibody group, they're 3 in each category, a low, middle, and higher. Of course, these are 3 logs below the ones that have been previously exposed.
And then do you know the kinetics when you expect to see the lowest level
so
of
in
antibodies?
In myasthenia, there are effects even after 2 doses. So we think probably the majority of the effect will be seen early. But we've been intending to use the usual induction dose of 4 weekly doses.
And Giuseppe, sort of same question. Are you going to go low?
The challenge has been that it is not widely used in younger children. And so some of the PK data that drives dosing is mostly in late school age adolescent and not in the elimination seems to be higher in younger kids. So we may not really know the dose to give it to the youngest children. So we're going to bias towards the older individuals where our Genix has actual PK data and not just modeled the PK.
Yeah, on my side, we are going to start with the lowest titers, of course, also for safety reasons, because we still don't know what's going to happen after the co-treatment with the IDES and the vector in an individual which is seropositive. It's, just to be on the safe side. So this will be tighter around, higher than what they are. But now we are including at less than 1:1, so it's almost no tighter. Mm-hmm. And there will be 1 to 10, I think, and higher, depending on how the first patient works, basically.
Are you setting an upper limit that you're not going to go higher than?
Well, the upper limit is not dissimilar to what I was showing in primates.
400.
Yeah, I think this is at least for now. We have a part of the study is, of course, just to see the efficacy and the things, but we are also doing some other analysis on the samples we will get. So we'll probably, depending on those analyses, we'll decide whether we are confident that it's going to work even in highest titer. Because there is one thing which is, for me, is still not unanswered in humans, is if the FAB2 fragments are really neutralizing. So this is the first question we're going to answer. So whether if we do see efficacy, then we can go ahead and see, but this really depends on...
So you don't know if the FAB2 fragments are cleared?
They are partially cleared, but they still remain. Part of the neutralizing and resistance power that we see is really that. But the thing is that this is in vitro.
Right.
so
relatedly, does IDES do anything to IgM?
Look, at that stage, we don't see any IgM. We have measured that. I mean, after injection with the IDS, we haven't seen any IgM going up.
No, I meant it doesn't cleave IgM.
No, it doesn't. It doesn't.
Okay.
It's true that, at least in our experience, but you were making a comment about that. We don't see much IgM, but it's true that for what you were saying, I mean, maybe you are masking some IgM.
And to that note, especially in your rabbits, assuming that you also get no cleavage of rabbit IgM, it might be a good model system in which to interrogate the relevance of the anti-V IgMs relative to IgGs.
Okay, because they mount super high response, right? That's the thing, yeah.
So related to IgM response to IDES, though, I mean, what is the potential to be able to just give IDES, instead of just one dose of IDES, 2 doses of IDES, because maybe you'll get those really high antibody titers completely to 0 if you continue to dose it. But I'm assuming there's some sort of IgM response that limits that. Is that right?
Possibly. Frankly, for IDS, the risk is more an anaphylactic shock because it's a bacterial protein. So I would not really try to dose many times. There are new molecules, new IDS that are developed specifically for that to enable redosing. But there is another issue for me, which is related to the affinity. So for me, there is anyway the difficulty for IDS to cap the antibody under a certain levels because of the affinity. So probably it's there where you can really get some, better IDS in that sense.
So there's a ceiling, you think, as to the effect?
It could be. Yeah.
So what's the concern about just dosing higher with IDS?
No, higher you can do. Higher you can do. Well, in
Repeated
monkey, in humans you cannot do that high. But higher is not really changing completely the situation because it's once again for me it's a matter of affinity. So
within one session, basically. But to your point, if you could continually lower the antibody levels by avoiding the anti-drug antibodies against IDES, this might be even more useful. And in fact, when it's used in renal transplantation in conjunction with other immunosuppressants, the adverse event rate is much lower. It's much more well tolerated. So it could be used in conjunction with rituximab.
Yeah.
There's no more questions. I think we can go for coffee. And at what time should we all be back?
Yes.
3:45. Thank you.